Welcome to the lecture of DNA damage repair regarding repairing strand breaks. So students, we will discuss about the single stranded break repair regarding SOS repair, regarding recombination repair and translesional DNA synthesis. We will talk about double stranded DNA break repair regarding non-homologous end joining method, regarding microhomology mediated end joining and homologous end joining regarded by recombination repair. So we know that the single stranded break is not creating any larger damage in the DNA because bases are chemically modified and changed, so it can create some sort of mutation and that can be repaired easily. But double stranded breaks actually give rise to the lethality to the cell. Okay, so it can create damage to the cell which ultimately leads the cell towards the death. So we have to know. So DNA strand breaks have two types: double stranded breaks as well as single stranded breaks. When a particular replication fork is moving and encountering a single stranded break, okay, we know that DNA polymerase 3 has the editing property. Fine. Suppose when it encounters a gap, okay, then it can reinitiate by having the primosome structure, having pri A, pri B, din A, B activity where DNA polymerase 2 and polymerase 3 can take part. Otherwise, if it see some sort of single stranded gap, then it stalls the replication over there, creating a double stranded break, which in turn will be repaired by several post dimer initiation strategies like recombination process. Okay. Now what is the fate of replication fork? Suppose if thymine-thymine dimer is present and DNA polymerase 3 has some sort of editing capacity, so against first thymine it will add adenine by Watson-Crick base pairing. But when it tries to add the second adenine against the thymine of the thymine-thymine dimer, it will add them at the syn configuration by Hoogstin base pairing, which creates some of the distortion in the helix structure. Okay, so again it will remove that particular adenine by editing function. Again the cycle will carry on. Now Rec A is a protein which can bind to DNA polymerase 3 and actually it inhibits the editing function of the DNA polymerase 3 and helps to bypass that particular lesion, okay, by creating a gap or by inserting any random bases, okay. And sometimes Rec A by PCNA class switching, polymerase switching activity can recruit the translesional polymerases, okay. So that is the fate of replication fork when it encounters some sort of lesion or gap in the single stranded DNA of the double stranded version. So what is going on? I have already talked about the reinitiation, okay. So when we are talking about this sort of reinitiation, it includes the post dimer initiation as well as trans dimer synthesis. Post dimer initiation includes the recombination repair as well as regression, okay, and this sort of trans dimer synthesis, it includes the SOS repair as well as translesional DNA synthesis.
Ma'am, what is post replication repair?
Very good question. So the replication has made already a single stranded gap, okay. So the post replication repair is the damage tolerance strategy. So it can be done by post dimer initiation against lesion or gap. So remember about the lesion made by the thymine-thymine dimer, as well as trans dimer synthesis, that is the synthesis against a lesion that have already present in the mother strand, okay. It follows regression as well as recombination as well as translesional synthesis. But in case of recombination, somehow some chromosomal aberration may happen. okay, but regression is errorless. In case of translational synthesis, we can see the targeted as well as untargeted mutagenesis also. So we are talking about the post-dimer initiation processes, okay. So it includes what? Number 1, regression, and number 2, recombination process. So regression is a type of recombination. So what we can see, when a lesion is found, that will be bypassed by regression model, okay. But when a gap is made in the single-stranded DNA of the double helix, then the recombination will come. Let's first talk about the regression model, okay. Suppose a lesion is present on the mother strand, okay. Now newly synthesized strand is passing. Now when it encounters a particular DNA lesion, okay, so how to overcome that? Now already some lagging strand is made. That particular lagging strand will be taken as template to synthesis and to bypass the lesion present in the original mother strand. In prokaryotes, that is mediated by REC-F-O-R pathway, okay, and this REC-F-O-R, with the help of REC-J also, they recruit the REC-A protein to solve this purpose, okay. And in case of eukaryotes, that particular regression, that is the lesion bypass, is done by several endonucleases like XPF and ERCC1, which ultimately give rise to the interaction with BRAC1 with RAD51, the homologous protein, as REC-A protein in prokaryotes, okay. So in case of prokaryote, this sort of regression is giving rise to the 3 helical chicken foot model, okay, by which the lesion that is already present in the mother strand can be easily bypassed automatically. Next, we will talk about the recombination repair, okay. So recombination repair is actually operated against the single-stranded nick present in the double helix, okay. So we will first talk in case about prokaryote strategy, then we will talk about in case of eukaryotic strategy. Suppose the replication fork is proceeding. When it encounters a single-stranded gap, okay, then and then they will stop by creating a double-stranded blunt end. Fine. Now these double-stranded end will be targeted by REC-BCD enzyme. This REC-BCD enzyme have the several multiple function. REC-B has the helicase activity, REC-C have chi-site recognition activity, I will talk about that, and REC-D have some exonuclease activity. So by helicase activity, it will unwind the DNA from the double-stranded end, fine, and by this sort of exonuclease activity, it will start to chew up from the end, and it will first chew up the 3' ending end faster than that of the 5' ending end, okay, and it will encounter a particular sequence called chi sequence, GC, TGG, TGG from 5' to 3' end, okay. When it will encounter such chi sequence, we can find 5 times, 10 times of these chi sequence within the 100 base pair in case of E. coli. When they encounter such chi sequence, they stop their cleavage, okay. But the faster way, it will cleave the 5' end for the production of 3' hydroxyl overhang, okay. Now these REC-BCD will recruit REC-A protein to wrap around the cooperative fashion over the 3' hydroxyl overhang. Now this REC-A is a master protein, fine. So it has some sort of ATP dependent helicase activity, fine. Now this REC-A, after winding up the 3' hydroxyl end, it drives that particular end for strand invasion towards the sister chromatid, okay. So it will displace the strand and it will make some sort of 3 helical structure along with the double helical structure, fine. Now when they will perform the strand invasion, a particular chi site is getting produced, fine. So that particular chi site is called as branch. That branch will be recognized by REUV-A protein. REUV-A protein will recruit REUV-B protein, fine. It has some sort of ATP dependent helicase activity, fine. So it will be needed for the branch migration. And by DNA polymerase 3, again the synthesis can be proceeded just to overcome the gap, fine. And after this sort of chi production, the branch production, branch migration, the model will be, the branch will be resolved by another protein, that is the endonuclease activity of RUF-C protein. Okay, when the branch will be resolved, they will be giving rise to the recombined product where no gap is present. Okay, so I have already discussed about a model that is called holiday model. Okay, and all the isomers are called holiday intermediates, and the chi site is called holiday junction. So single stranded nick, fine, that give rise to double stranded end, then strand inversion, branch migration and resolution of the holiday intermediates that actually help single stranded gap repair in case of prokaryotic DNA. Okay, and we can find in case of eukaryotes that is mediated by RAD51, which is homologous to the REK protein in prokaryotes. Okay, and in mammalian system you also find RAD51. In downstream they are reacting with BRAC1 and BRAC2, which are very responsible when they are getting damaged for the breast and ovarian cancer. Okay. Next, transdimer synthesis, fine. So already a gap, if it is getting produced and if it creates lethality to some of the coding genes, so that must be repaired by some emergency purpose genes. Okay, so they must obtain some procedure as SOS, emergency repair system, or translational DNA synthesis. Okay, SOS repair, emergency repair, fine. So already a single stranded gap is present, fine. Cell has to repair that particular gap. What is going on when such single stranded DNA is getting produced by damage, single stranded break, that will be recognized by REK-A. So single stranded DNA along with REK-A that can activate the coprotease activity of the REK-A, which actually targets some other protein called as LEGZ-A. LEGZ-A has the target site for the proteolytic cleavage, and by autoproteolysis the LEGZ-A will be cleaved. So what is the function of LEGZ-A? LEGZ-A is the repressor over some of the important genes, fine. Now LEGZ-A acts as repressor against, it sits over some consensus DNA sequence called as the SOS-box sequence, that is 20 base pair sequence over several genes, and the LEGZ-A repression affinity changes from gene to gene. There are 43 genes, or there are sometimes 20 genes under this regulon of the SOS. So that is called SOS regulon. When LEGZ-A is getting cleaved, so it turns out from the repression, fine, from the blockage. First, it relieves the REK-A and LEGZ-A synthesis of itself, fine. But once LEGZ-A is getting produced, they will be automatically cleaved because already activated REK-A is present, which is bound to damaged single-stranded DNA, fine. Next, it will relieve some of the excision repair genes like UVR A, B, C, D. They will go and they will repair the thymine-thymine dimer lesion present in the mother strand, which actually is giving rise to the single-stranded gap, fine. Next, it will relieve some of the gene like UMUC, UMUD. Now, UMUD after synthesis will be cleaved to produce UMUD prime, fine. Now, 2 UMUD prime along with C, that is UMUD2 prime C. Okay, so this UMUD2 prime C is called as mutasome. It will bind to the single-stranded break. Okay, so Reke will recruit this U mu D2 prime C as translesional polymerase called as polymerase 5, fine, by eliminating DNA polymerase 3. So they can insert any base, especially against apurinic site, it will insert A and they can fill the gap. Okay, so they are some sort of translesional polymerases. Other than U mu D2 prime C, DINB having this sort of polymerase 4 activity which is much more errorless, they can synthesize the gap once again. So not DINB, SOS can also helps to synthesize SAL A, SAL B, they are all cell division or cell cycle regulator because cell must be repaired before cell division occurs. Okay, Rec N, Rec U, there are so many other proteins will also be synthesized, fine. But once the single-stranded gap is repaired, then again the double-stranded DNA will be produced. So no more Rec will be activated, so no more Lex will be autoproteolized. Again Lex will be dimerized and it will act as repressor over the source regulon genes. Okay, so in this way we can find there are so many translesional error prone polymerases are getting synthesized to join the gap as emergency. Translesional synthesis in case of prokaryote that I have already discussed, DINB, that is like polymerase 4. Okay, they give rise to the errorless repair, fine. But U mu D2 prime C, they act as polymerase 5, they give the error prone repair, fine. Now the same thing can also be possible in case of eukaryotes. The normal polymerases are there like polymerase alpha, delta, etc. But when such single-stranded gap is encountered, then polymerase switching is happened, okay, by the help of pCNA. Okay, so from DNA polymerase, normal DNA polymerases, that will be switched towards the polymerase eta, polymerase tau, zeta, etc. Polymerase eta is more or less error free, but polymerase tau fixes the damage. Okay, but if it encounters any mismatch, that can be extended by polymerase zeta. So in this way, differential type of DNA polymerases can be found which are called as translational polymerase. So remember student, though this is very emergency purpose needed action, that is translational synthesis, but they are very much error prone. Okay, they can introduce the mutation in the daughter strand, but they can save the lethality. Okay, that is very much needed for a particular cell. Double-stranded break repair. First, NHEJ, non-homologous end joining. It is actually found in case of eukaryotes, but in case of prokaryote, you can also find the presence of Q protein along with a multifunctional LIGD protein having ligase, polymerase as well as the nuclease activity. Okay, so this actually helps to repair the double-stranded end. You know that when the double-stranded ends are produced, that is very lethal for the system because the cross linkage can give rise to the translocation. So when they will be repaired, they will be handled with care. Okay, so let's talk about the NHEJ mechanism in eukaryotic system. There is a protein called Q protein which is heterodimer of Q70 and Q80 protein. So Q protein, over there two binding side, they will recognize the double-stranded broken ends on their either side. Okay, so when they will encounter such damage, they will recruit the downstream protein, that is DNA protein kinase CS. Fine. When this sort of protein kinase is activated, it will recruit XRCC1, okay, and XRCC4, and they will ultimately recruit the DNA ligase 4 to join the ends. Okay, so we are not finding any template, fine, but it is getting synthesized and joined. Usually Pol mu is taking part during this sort of NHEJ. Next, MMEJ mechanism. When the NHEJ mechanism is not available, that during the intra-S phase of cell cycle, because at the late phase we can find the homologous recombination happens. In the early phase, non-homologous enjoining process happens, and at intraphase this sort of MMEJ happens when the NATJ is not available. So there is 5 to 25 consensus complementary sequence over the two broken ends of DNA. Okay, so they will try to make the hydrogen bondings, fine, by making the flaps. That particular flap will be eliminated. Okay, when that particular flap will be eliminated, then polymerase will synthesize the gap. Okay, and they ultimately will be joined by some sort of ligase. Now we can see that is already found in case of eukaryote, but in case of prokaryote we can find the similarity, the homologous in case of DNA polymerase 1 gene with respect to the Drosophila gene regarding this MMEJ repair. Next we will talk about the double-stranded break joining by homologous recombination. That is actually the main repair system for double-stranded break repair. That is more or less errorless with respect to NHEJ or MMEJ. Let's first see the mechanism of double-stranded recombination repair in case of prokaryotes. Okay, let's talk about the steps involved individually. So what is going on? The double-stranded breaks are made. Okay, now on both the strand, again the 3' overhang will be produced by the presence of the RegBCD enzyme. That particular 3' overhang will run towards the sister chromosome homologue. Okay, and they will form two branches, that is called duplex. Okay, when they will invade the strand, fine, and when they will make such branch over there. Again the branch migration will be happen for the sealing of the gaps by the synthesis of the DNA polymerases, and when the synthesis will be ended up, then resolution of such branches, that is the holiday model intermediate, will be resolved by RUV ABC, which gives rise to the ultimate recombined product where double-stranded breaks will be repaired. In case of prokaryote, you can find the participation of RegBCD and Reg-A protein both in case of single-stranded break as well as double-stranded break. The significant thing is that in case of double-stranded break, already the double-stranded ends are created that will be targeted by the RegBCD. But in case of single-stranded break, when it will be encountered by the next set of replication by the replication fork, then only the double-stranded ends will be created, which will be targeted by your RegBCD. In case of eukaryote, you can also find this sort of double-stranded break repair by homologous end joining. Okay, but the thing is that the protein components are very different. Okay, suppose we can find the Reg-A like protein, that is RAD51, that also we can find in case of yeast cell also, and we can find BLM helicase having some sort of property like Reg-D helicase activity. Now Reg-B can interact with Reg-A protein. Okay, here we can also find some sort of interaction of the BLM helicase with RAD51. Okay, not only that, we can find some sort of complex structure is produced by DNA2, number 1, by RPA, replication protein A, along with BLM helicase. Ultimately, when such double-stranded breaks are produced, okay, that will be sensed by ATM kinase. Okay, so ATM is related to ataxia telangiectasia, okay, a disease. So ATM kinase will recognize the double-stranded break. Fine, ATR kinase that will sense the replication fork blockage. They will ultimately phosphorylate some sort of downstream protein like P53 as well as BRAC1. They are needed for cell cycle arrest as well as repair, and ultimately they will give rise to the synthesis of CH1 and CH2 kinases, which actually help the transcription of repair related gene as well as polymerase activity that is needed for your recombination purpose to seal the double-stranded break. Now this ATM and ATR, they are called the sensor. They ultimately give the effector molecule. Okay, so effector molecule will be activated first via adapter molecule like BRAC1, P53, which will actually give rise to the effector molecule like CH1 kinase, CH2 kinase. They help to propagate the PCNA, processivity factor, for polymerase switching. Okay, so in this way translational synthesis as well as recombination repair happen with respect to the double-stranded DNA break.